So if I can welcome Stephen up to the Evaluating Biopharma main stage here.
Good afternoon, Ben. Good afternoon, everybody, or good morning, as it might be in the US.
Stephen, how are you?
I'm very well, thank you. Thank you for having me today.
Yeah, thanks for being here. If you wouldn't mind, give us a little bit of background bio on yourself so everyone knows the expertise from which you'll be discussing your topic today.
So my name is Stephen Judd. I work for Arcadis DPS Group, was DPS Group until last week when we were acquired by Arcadis. So I work in the A&E company, so architectural engineering firm. So we do the facility design and the process design for manufacturing companies for the development of new facilities. I've around 18 years experience in the industry at this stage, and I've spent my entire career on this side of the fence as opposed to working for a manufacturing company. So my role for DPS is as our process SME for biologics and cell and gene therapy, and my focus is on the early design phases, so feasibility study, concept studies, and into basis of design, where the main process philosophies and facility design philosophies are developed ahead of the detailed design of the facilities. So I'm going to be talking today around the key factors associated with optimizing those facility designs.
Awesome. Well, thanks for being here, and let's just jump right in then. So maybe first explain to the audience the basic differences between an autologous and allogeneic cell therapy facility design. What might be different? Why might they result in different design considerations?
Okay, so this I'm probably here going to follow on from what some of what Jonathan and Marty have already said, but discuss how some of the things which they've said apply to the actual facility design consideration as opposed to the manufacturing science aspect.
Yeah,
so
great.
if we start with an autologous facility, so an autologous therapy is, as has been discussed, is a personalized medicine. So it is one batch equals one patient. That's not going to change. That is as you scale up the manufacturing capacity of your facility, that means scaling out. So you have multiple patient batches manufactured in parallel in order to increase your throughput for your facility. So scaling out versus scaling up typically means that you need a greater manufacturing footprint, so you need to look at, even though it's small scale, even though you're only talking about benchtop pieces of equipment, you still need to be able to operate everything in an ergonomic fashion. Everything still needs to be neat. You still need to be able to move around the equipment to be able to set up the kits in an effective manner. Therefore, and one of the key factors is ensuring chain of identity and avoiding mix-ups. So the layout of the facility needs to account for that, and the scale out factor either needs to have you need to look at ways that you can optimize the layout, optimize the footprint by potentially stacking pieces of equipment, but then you also need to ensure that you have the ability to be able to reach the equipment that you're working on and be able to operate these things in an ergonomic fashion. So one of the key factors associated with chain of identity and avoiding mix-ups is looking at ballroom suites. So you're manufacturing you have multiple patient batches in one open plan area. So a key factor there is early engagement with the regulatory authorities. So the regulatory authorities are going to look at and question how you avoid those mix-ups. And from an optimization of a facility design standpoint, you want to have the most amount of equipment possible in an open ballroom, as opposed to having to segregate between different areas. So early engagement with the manufacturing, with the regulatory authorities, helps to be able to bring them along through the design process, and that doesn't adversely affect the approval process and impact your speed to market when it comes to actual regulatory approval for that facility. The other factor is there was a lot talked about earlier about variability in the patient material, because you have obviously the viability of the cells from the patient is not the same as it would be from a healthy donor. One of the key factors that this affects is cell expansion. So if you do a cell expansion step, you're looking for a target cell density, and the cell expansion step completes after you achieve that cell density. But you may have a variability in your process, so it may take, say, between 3 and 8 days to achieve that cell density, and you sample to ensure that you get to that point. Where you have healthier material, that obviously achieves the cell density in a shorter period of time compared to the patient material that is not as viable. One of the critical factors is the longer the expansion phase, the longer the step, the more pieces of equipment you require in order to achieve a certain throughput, compared to you having shorter periods, because obviously you turn over the equipment more quickly when it comes to shorter cell expansion periods. So one of the critical challenges is ensuring that you implement the right number of pieces of equipment in order to achieve your throughput. One of the approaches to do this is to model your facility using statistical analysis associated with those cell expansion periods in order to effectively determine how much equipment you need. You don't want to be too conservative, because that adds cost and footprint, and you don't want to underestimate, because then you won't achieve your throughput. So this is one of the big challenges which is associated with those processes. When you look at the comparison then with an allogeneic process, as Marty was talking about there, you have the potential for scale up. You have the potential for, if you look at a smaller scale bioreactor system, which is going from, say, a 20, 50 litre stir tank reactor, compared to a 1 to 2 litre cell bag, which is associated with an autologous process. You can manufacture a much greater number of batches for a much smaller footprint. You have less variability in your material, because you have variability from donor to donor, but you can characterize the material better because it's coming from healthy people. And you don't have the same chain of identity and mix-up considerations, because your aim is to produce an off-the-shelf medicine that is going to be available to multiple people from individual donor material. So in general, if you look at an example of an autologous versus an allogeneic, so one example of this can be a CAR T. The allogeneic, the majority of I think maybe all of the therapies that are approved for CAR T at the moment are autologous, but there are allogeneic candidates in development. So the allogeneic candidates obviously need to ensure that there is no possibility of graft versus host, but the allogeneic therapies have that advantage for better efficiency, more streamlined processes, higher throughput with smaller footprints, which is a key factor in optimizing the space that you have available for manufacturing.
We had a question come in regarding this. Do manufacturers need to consider the modularity of the equipment when designing the facility, like open ballroom versus small rooms?
Yeah, so there's a lot of the equipment associated with, if we look at autologous manufacturing, there are different platforms associated with, for an autologous process, there are developments in advancements in equipment which allow multiple process steps to be performed on single pieces of equipment versus other processes which will involve individual pieces of kit for each individual process step. So as the advancements in technology progress, the processes are becoming more closed, they're becoming more automated, and this is allowing for process closure is something which I will come on to in a minute, but certainly the more closed, the more automated the systems, the more capability you have for ensuring that you can efficiently set those pieces of equipment out in a ballroom area.
Let's touch on that now then. That's a good point and a good segue, I think. So Chad, if you wouldn't mind, about the value of the closed process and what sorts of operational challenges may arise there, and also why avoiding manual operations is the way to go.
So in general, closed processing obviously means protection of the process from the environment and from other unit operations. So the closed processing allows segregation to be minimized. It allows you to be able to install multiple pieces of equipment in the same ballroom suite compared to having small rooms. Yep. The ballroom area minimizes the overall footprint. It reduces the number of rooms, number of airlocks, so it reduces the construction materials, it streamlines your schedules, and can help with increasing speed to market. The other major factor in closed processing is reduction in clean room grades. So a big thing that I would like to talk about here is the grade B versus grade C manufacturing environment. So why do we use grade? So a lot of facilities coming through will look at grade B suites for different manufacturing operations. Why do we use grade B suites? For protection of the product. In certain cases, because moving to an isolator compared to a biosafety cabinet means that you may lose some of that. The operators might not find it as intuitive for making those small manipulations, and the operability is perceived to be impacted. And then there's the other factor, which is speed to market. So a lot of times a product which is coming through development, the main focus is to get it to market as quickly as possible, which means changing the process as little as possible as you come through clinical trials to commercialization, and then maybe optimizing the process when you get to commercialization. But one of the key factors in modern facility design is also sustainability. Sustainability is a key factor in all new facility designs, and in fact it is a specific requirement, certainly in Europe, associated with designing a new facility. We have something in Europe called NZEB, which is the near zero energy building regulations, which basically means that when you're designing a new facility, you need to show that you're minimizing energy consumption. And then a lot of facilities look at things like LEED and BREEAM, which are sustainability goals in operating your facility. The single biggest energy user on a manufacturing facility such as this is your HVAC system. Your HVAC system is what conditions your air. This is what maintains the environment within those clean rooms. So to give you a comparison, a design phase air change per hour for grade B versus grade C is 65 air changes per hour at grade B versus 35 air changes per hour at grade C. So there's a massive difference in the energy consumption associated with operating those facilities. So when we look at closed processing, there is a lot of there is a significant level of equipment on the market today which allows closed process operations, whether that be closed tube contacts, so tube welding or aseptic connectors. So tube welding is not necessarily seen as the most robust approach, but it is a practical means of process closure. And then advances in aseptic connectors now have their connectors on the market that are down to 1/16 inch. So the small tubing associated with cell therapy operations can utilize these connectors. Having closed tube-to-tube connectors allows you to operate equipment in a closed manner in a grade C environment. And there are examples of these in facilities that I've been involved in designing which operate these processes in a closed environment. Where you have operations that can't be fully closed, such as I'd say adherent cell stacks or filling operations, then these can be effectively carried out in isolator technology. And there are examples of cell therapy, cell processing isolator offerings from certain isolator vendors, and then small scale filling systems that can be effectively operated in an isolator, which allow those isolators to be installed in a grade C environment, and that minimizes the clean room grade. Because the other thing associated with grade B is it's very challenging to maintain and onerous to work in, especially if you look at the sanitization requirements associated with bringing material into those suites and the risk that that creates to environmental contamination of the area.
Right. Now you have a case study, Stephen, regarding a multi-product autologous CAR T facility in Europe. Would you mind jumping into that?
So the one of the main considerations associated with the ATMP regulations within Europe, and I think they're the same with the FDA, are that different products, once they include the genetic modification, so once they include the lentiviral vector, the different products need to be segregated, but other operations outside of that don't involve that genetic manipulation don't need to be segregated. So when you're looking at a multi-product facility, it's important to break it down into the different areas. So what operations can you take, what say steps prior to the transduction step can you take and put into a common area, which allows you to use allows you to have a common ballroom space where you have operations associated with multiple products being carried out in the one space. And then once you move into your operations where the transduction step is carried out, once you introduce the viral vector, then you need to have separate suites for different products. So it's how do you manage those operations to best kind of split up your manufacturing operations so that you can use different ballroom areas in order to best lay out your facility. You have it's important to have unidirectional flow through manufacturing suites in order to prevent your the cross-contamination associated with people entering and leaving a manufacturing area. It's important to separate your product and waste flows, but all of that can be contained within a manufacturing suite. And as long as you ensure that everything is properly sanitized before you move it out of the manufacturing area, then having bidirectional circulation spaces can help to streamline your overall space, as opposed to having supply and return corridors, which can lead to a greater footprint. So how you operate the facility, how you manage your procedures, how you handle your materials, how you handle your waste, all of that needs to be, or if your operational procedures are effective, then you are able to streamline those areas. In the case study presentation, the majority of the operating facility was grade C because it is closed process operations. It is effective process closure of single-use systems which are able to operate in a grade C environment. Some of the key things that are a challenge for autologous therapies in particular are around warehousing, logistics, cold chain, freezer chain, that sort of thing. So you have small scale equipment, but you have a lot of it, which is a lot of different pieces of equipment operating in parallel in order to achieve the throughput. So your management of your single-use strategy is very important. What is your business case for the quantities of single-use consumables that you keep on site? Do you engage with a third-party logistics company in order for that company to have an extra 2 or 3 months worth of storage, because single-use consumable lead times are going through the roof at the moment? So that's very important. And then the consumables kits, you have that specialist piece of equipment, which is your cell therapy system. But then you have other pieces of equipment like bags and connectors and other things which come from other vendors, which are part of that overall kit for your unit operation. So in order to have efficient operation, that kit needs to be assembled ahead of time, so it's there available for the operator when they actually when they are looking to set up their piece of equipment. They don't have to go looking for different components in different areas. So an efficient means of managing how that is assembled, an efficient means of managing how all that is sanitized, so that you streamline your operations for bringing it into the suite, is very important. So having a dedicated kitting area, which is of the equivalent grade to the manufacturing suite, where you're able to pre-sanitize everything, and then you minimize what you need to do when you're actually bringing it into the suite. That's very important. And then when it comes to cold chains and freezer chains, it is one of the challenges is you may have extended periods of product release. So your QC and your QC product release testing may take between 2 and 4 weeks. During that time, you have a lot of other material within the facility which is backing up and needs to be staged in freezers. So your freezer capacity is a critical consideration. And then you look at different means of freezer design. So you've got freezer types which are manually operated. People are manually taking the particular sample from a particular slot within that freezer, or you have new advances in freezer technology which have automated retrieval, which gives you an improved safety because people don't have to actually go into the freezer, and improved chain of identity because everything is automated. But then those freezers have a lower capacity than the manually operated freezers, so it's a trade-off, and you need to look at space that is associated with being able to set those out. So that is definitely a critical factor when it comes to setting out that area and ensuring that you have enough space for all the freezers that you need.
Hmm. Yeah, there's a lot to consider there. My lightning round final question for you, in considering a lot of that which you mentioned, are you building some of these new intricacies, like the longer supply chain lead times for single use, or changes in technologies, freezers, et cetera, into your statistical modeling for your designs to better reflect reality? Or are you keeping your designs the same, and the total variance of reality just tends to stack up?
Well, the statistical analysis for the variation in the batch process step times, really that aspect of it comes from the client, really, because it comes from the model of they have their data of obviously the patient
Yeah.
aspect. And that is we need to get the best available information to build that into the model. But just quickly, actually, I know we're a little bit running, but one thing that I just would like to highlight quickly when we talk
Please. Yeah.
about market pressures and extended lead times is, if you look at CAR-T in particular, they're one of you have two precursors to the CAR-T. You have the lentiviral vector, and you have if you're using transient transfection to manufacture the lentiviral vector, you have the plasmids, which need to be part of that process to make that part of the CAR-T process. And with the way that the market is going, there is a lot, those supply chains are getting very, very pressurized. They're getting longer lead times, higher costs. So I think if the company has the capability to do it, looking at bringing those manufacturing processes in-house is ultimately going to protect the supply chain and reduce the cost of goods for the overall process. Now you're looking at an E. coli based fermentation for your plasmids, followed by a mammalian cell culture based process for your lentiviral vector, followed by your drug product for your CAR-T. But provided you have the right level of segregation, there's nothing stopping. There's no reason why you can't have those defined manufacturing facilities at the same site, as long as you have the right levels of segregation between the different areas. So these are definitely things that need to be considered, because ultimately they are things which can, if you look at, say, CAR-T as an example, the availability of the precursor material is something that can significantly impact the ability to supply that medicine to the market.
Yes. Well said.
Yeah.
Stephen, thank you so much for your time and insight here today and sharing with our audience. To the participants, we're going to jump next into the deep dive network session, where you can ask questions of the panelists, Jonathan, Marty, and Stephen, from the sessions today. On behalf of Brian, Kane, and the team, I thank you for attending and participating, and we'll speak soon at the switchover.